I know I'm standing in the way between us and lunch, and my whole objective here is not to get the hook and be very efficient with the disclosure of the data on nChroma Bio. But many of you may know that nChroma is a product of two companies coming together at the end of last year. We merged two companies together to form nChroma Bio, and this is really the first opportunity for us, after launching the new co in January, giving an intro to the company. So that led to this profile, a pipeline of potent, durable, and selective epigenetic silencers. That is a foundational technology for the company, with an amazing team, with deep experience not only on the research side of genomic medicine, the development side, but also importantly, as we hear throughout the conference, on the manufacturing side, leveraging LNPs on the manufacturing side of things. We have a lead program heading into the clinic very shortly, with CTAs being filed in Q4 in multiple jurisdictions for hepatitis B. That's predominantly what I'll talk about here today. Pipeline of targeted in vivo therapeutics behind that, leveraging the novel cargo I just described, but also novel delivery technologies. An amazing investor base and capital to take a run at it, which is a nice thing in this environment to say. So an incredible progress over the last 15 years. We've heard about it. We just heard it from a number of speakers here. Excited about the progress, but we also know, and we're talking here, about the many challenges remain for broader adoption, and not just in rare diseases, but also in larger markets. I'm incredibly bullish about the prospect of in vivo genomic medicine right now. I feel like we're on the cusp of a massive innovation curve, and the innovation's not only on the clinical side and the science side, but also on the commercial side. We believe that epigenetic silencers are one of the tools to allow us to get there. It leverages a natural mechanism of silencing gene expression. That means that we do not cut, we do not nick, importantly. In vivo, leveraging existing LNP technologies, highly selective, durable, potent, and again, importantly, from the commercial side, we believe with an mRNA-delivered LNP, or an LNP-delivered mRNA product, we have an opportunity to make a very, very compelling case with attractive COGS in the markets we're pursuing. So what is this mechanism, if you're not familiar with it, that allows for natural silencing of gene expression that we're taking advantage of? Top left of this, you see an open, active, and accessible gene, and it's a process of methylation that naturally occurs, which closes the gene, renders it inactive, inaccessible, and therefore silenced. The small little character you see in the green, blue, and red is a depiction of our epigenetic silencers that have multiple components that allow for that selective, durable, and potent effect. So look at the pipeline. I'm really only going to spend time on the Chroma 1001 HBV program, but if I have time at the end, we'll hit the rest of the pipeline. It's an epigenetic silencer using LNP, again, in the CTA filing status. So what is Chroma 1001? If you look at the left side of this screen, it's an LNP-delivered product, mRNA-based epigenetic silencer with a guide RNA. So leveraging a lot of the technologies that are standard today. Obviously, the epigenetic silencer itself is something we've been working on for a long time, and that is unique. I'll describe why it's uniquely suited for hepatitis B in particular. And really, that's driven by the fact that it permanently silences both forms of viral DNA, a very, very important characteristic for hepatitis B in particular. Mentioned it as potent and specific, with the expectation that we will see a broad therapeutic window. Obviously, we have to prove that in the clinic. And importantly, without the risk of genotoxicity associated with other products like cutters and those that nick the DNA. I'll get into some of the reasons for why we think this is a perfect therapy for hepatitis B, but the goal here is clear, which is durable functional cures for this population with a single course of treatment. So, hepatitis B. Big problem, big challenge, and a disease we have not solved, despite the fact that there are vaccines and therapeutics available. These numbers tell the story. I've seen as high as 300 million population of chronic hepatitis B patients across the world. 1.2 million infections annually. Clearly, we have a long way to go, and unfortunately leading to 1.1 million hepatitis-related deaths. Again, The challenge is clear. It's functional cures. So I want to take a minute and just get you familiar with the hepatitis B community. We've spent a fair amount of time with the Hepatitis B Foundation. You can read the quotes up here. Several of these individuals, patients, actually gave us time at our Boston office to give us a sense of what they're experiencing. And I wanted to read you a few quotes from them, from our visit, which really brings it home. Years of pain and escapism, worry about dying at a very young age, and its impact on everyone I care about in life. Consider HBV in almost every decision I make in life. There is no taking days off. And every appointment I go to, I carry an anxiety about viral load levels with me. You get a real sense of the lives that these patients live, and they're looking for hope because current therapies just aren't doing it for them. So what does an ideal product profile look like? You can see it here. In an ideal world, and this is aspirational for sure, single course, durable, hopefully for the rest of the patient's life, so they have to be off treatment, delivering functional cures, as I referenced, leading to long-term reductions in cirrhosis and liver cancer and ultimately death, a commercial profile that is viable and competitive, and ultimately taking the stigma out of the disease that they live with every day and the challenge of lifelong treatment. You can see the construct I described on the right, which is an LNP-delivered technology, again, with mRNA-based epigenetic silencers. So take a quick step back for a second and talk about why is chroma 1001 ideally suited for the pathophysiology of hepatitis B. This is very important. There are two pathways that lead to viral infection. On the left-hand side, you see cccDNA. This is effectively a factory setting up in your liver to produce these virions that ultimately lead to HBV DNA circulating, infecting other cells, and S antigen production, which is immunosuppressive. On the right-hand side, you see integrating DNA, another pathophysiology of HBV. That leads to these HBs antigens circulating, non-viral, but circulating, and they are immunosuppressive. So you've got this factory set up in the liver that is producing more and more HBV DNA, infecting cells, and an immune system that's incapacitated. You can see why it's such an infectious disease. And the therapies that are out there today don't accurately and appropriately hit both pathways in a way that can lead to meaningful functional cures. So I'll hit this quickly. This is just an attempt to show the differences from a pathophysiology and mechanism perspective of why different therapies work differently. So if you look at the top left, light color is S antigen, dark color is HBV DNA, and the dosing of the single arrows down. Nucleosides are a standard practice in HBV. You can see that they're dosed very frequently, but when you come off, disease comes back, and unfortunately, nucleosides only hit one of the two pathways. For the ASOs and siRNAs that are out there, and certainly in the clinic, there is encouragement. You spread out the dosing, which we know that to be a part of certainly those modalities. But what's also true of those modalities is in the box to the right, that at the stopping of treatment, the infected cells, because they still are infected, are functionally active. And ultimately, you get the virions, the DNA, the antigens being reproduced, and the disease burden then overwhelms the immune system again, and the infection starts again. Functional cures demand that you come off therapy and have 6 months free of S antigen and HBV DNA. They don't afford that. There is some encouraging data, but they're not getting to where this community needs it to get to. And that's where chroma 1001 and epigenetic silencers come in. As you can see, one dose, durable effect. I'll show you some of the data supporting that. Permanently inactivates HBV DNA across both pathways, and that will result ultimately in minimal residual disease, as we won't be able to hit every hepatocyte, for the immune system to reconstitute and do its job that it was intended to do, but for the immunosuppression. So enabling functional cures. That's our goal. There are two established models here that I'll go through both of them and quickly hit the data, intended to mimic both pathways. So this is an AAV HBV model infecting the mice, intended to imitate the cccDNA pathway. As you can see from this data, the marker of S antigen in circulation on the left hand side, the y-axis, and days post single dose administration. Very dose dependent and getting a profound effect on S antigen loss. And I'll talk about loss in a second, but at 0.5 mg per kg, you're getting 2 log reduction. At 2 mg per kg, you're getting 3.5 log reduction. So in this same model, we tested 3 doses rather than 1 dose. And same structure here in terms of the chart, 1 mg per kg, single dose versus 1 mg per kg at 3 doses. And you can see the difference from 2.2 log reduction to almost 4 log reduction. And importantly, almost all of these mice were below the level of detection of S antigen. So that is obviously incredibly encouraging, and you can see the durability of this as well after a single dose. In the other established model of transgenic HBV, we're inserting 2 copies into the mouse genome, intended to mimic the integrating DNA, and you can see the same effect. You're seeing at 0.5 mg per kg, again, approximately a little more than 2 log reduction, and at 2 mg per kg, you're getting more than 3 logs reduction. This is the basis upon which we're really excited about getting into the clinic and testing this in patients. So here's where we are. Many years of optimization of these epigenetic silencers, academically and then in the company, showing a profile of durability, potency you just saw in mice, but all of the CTA-enabling work that we've done corroborates that, both in NHP models of putting these epigenetic silencers in for potency, durability, safety, as well as all the in vitro data as well. So we're in the process of filing CTAs this quarter, multiple of them, with a highly engaged and committed both patient community, as you saw, but also clinical community. It's exciting to see what the prospect of epigenetic silencers in the clinic. And we're on track for that first patient to be dosed early next year. Here's a brief look at the expected, or the first-in-human study, expected to be more potent than competitors based on preclinical data that we have and we've seen. We're treating chronic HBV-infected patients that are on nucleoside therapy. Eventually, the goal is to take them off, as I described. Single and multiple ascending doses, as you would expect from the data. GMP material ready to go. Strong KOL network, and hopefully dose that first patient early next year. So the pipeline behind that, I'll spend 2 seconds on that. We're not done. So if you look at the top left, we've got next generation silencers that are proving to be 2 to 3 times more potent than Chroma 1001, which may afford us the opportunity to have a next gen program to hit even broader populations. We've licensed in exclusively this CHARM epigenetic silencer, so you see that referenced on the top right. And we've seen potency in NHP targeting PCSK9 at levels for saturating silencing that I haven't seen anybody publish on. We believe it's at least 3 times more potent than the data that are out there, including companies like Verve. So incredibly potent silencer. It's both potent and truncated. And the truncated version of it is being pursued in CNS disease because we can fit it into a single AAV. There's proof of concept in a model of prion disease, mouse model of prion disease, which showed a very dramatic effect of an AAV-delivered CHARM epigenetic silencer. And so we are now embarking on and generating data in other indications that have broader potential reach, Huntington's, Parkinson's being examples of that. And we continue to invest in our EVLB technology for extrahepatic delivery. So an emerging pipeline that unfortunately I don't have a chance to really get into today. An amazing team with deep experience, as I mentioned, on the research development and manufacturing side. A great investor base that is a product of the 2 companies coming together. It's been incredibly supportive, including the capital raise at the end of last year. And we're heading to the clinic to be able to test our very exciting Chroma 1001 in hepatitis B patients with more to come beyond that. So thank you very much.
Thank you, nChroma Bio.